Crispr Cas9. How it's used. This is Dovey, and for my video, it talked about base editors. In addition to Crispr Cas9, there's base editors. Base editors can be more efficient and cleaner than Crispr Cas9 for making point mutations, offering advantages in correcting genetic diseases. When you look at them side by side, you can see that base editors are more like pencils that directly change one base to a different base. You can also find that the efficiency for making point mutations at target positions is much greater for base editors than for using Crispr Cas9, and it is much cleaner. Base editors allow significant advantage if your only goal is to change one base pair to another base pair.
Hi, this is Bella, and in a healthy rat, the dystrophin gene makes a full-length protein that keeps muscles strong, but in this rat, the gene had a broken section, so it couldn't make the protein properly, and its muscles were weak. Using CRISPR-Cas9, scientists removed the broken part of the gene, and now the rat can make a shorter but functional protein, and its muscles are much stronger, and the sad rat is happy again.
How it works. This is Dovey, and the first step is targeting. Cas9 is a type of enzyme that cleaves DNA. It recognizes and binds to a 3-nucleotide sequence motif called PAM. Scientists synthesize the guide RNA to contain a sequence of about 20 nucleotides that matches a particular sequence in a cell's DNA that they want to target. Once the guide RNA is added to Cas9, it will guide Cas9 to the target sequence. The target sequence can be nearly any sequence as long as it occurs near a PAM motif. It can be part of a gene's coding region or a regulatory sequence that scientists want to change in some way. Step 2 is binding. Once it binds to a PAM motif, Cas9 unwinds the DNA double helix. If the DNA at the location perfectly matches a sequence of about 20 nucleotides within the guide RNA, the DNA and matching RNA will bind through complementary base pairing.
Once the guide RNA pairs with its complementary DNA sequence, this pairing triggers Cas9 to change its three-dimensional structure. This structural shift activates Cas9's nuclease activity, allowing it to cut the DNA. Cas9 cleaves both strands of the DNA at precise locations just upstream of the PAM sequence, a short DNA motif required for Cas9 to bind and cut. The cutting action leaves a double-strand break, which is the first step towards removing or editing the targeted section of DNA. This is Bella, and the last step in DNA repair involves the cell's natural machinery fixing the break in the DNA. After CRISPR-Cas9 cuts the DNA, the cell works to rejoin the two ends, either by directly reconnecting them or by using a template to fill in any missing information. This repair restores the gene, allowing it to function properly once again.
Non-homologous end joining. Non-homologous end joining is the more frequently used, faster repair mechanism because the cell does not use a template to join broken DNA ends together. It is, however, an error-prone process that can introduce mutations in the target sequence. Errors are rare, but when the break is repaired correctly, Cas9 will once again recognize the target sequence and cleave it. Repeated cycles of cleavage and repair eventually result in a mutation. The type of mutation is random, but it will occur precisely within the desired target sequence. If the target sequence is within a gene's coding region, the mutation will likely inactivate that gene. The second type of repair mechanism is homology-directed repair, which is less error-prone and uses a homologous DNA template to accurately repair the break. Scientists can manipulate this repair system by introducing into the cell an excess of a DNA repair template along with the Cas9 guide RNA complex. The cell's repair machinery will be tricked into using the repair template to fix the break by HDR. By designing different repair templates, scientists can change the target DNA sequence into a new sequence. These templates could also correct an existing mutation by replacing it with a non-mutated sequence of DNA.
This is Bella, and one question I still have is what happens to the cut out piece of DNA after CRISPR-Cas9 removes it?